Right. Good morning. Thank you very much for the invitation. This is an excellent meeting. I've been to the American Society of Human Genetics for a number of years, never attended this meeting, and this is excellent. So what I'd like to do today is to give an overview of some of the genetics of infertility, and we'll discuss the genetic etiology and testing in different levels. So arbitrarily, I've divided patient categorization into hypogonadal, low levels of estrogen, and eugonadal with normal levels. And as we'll see with both hypo and hypergonadotropic hypogonadism, there's more is known about the genetics. In the eugonadal patients, I've listed several disorders here, and you'll see the first two have asterisks that I've called hypergonadal in the abstract. I mean, it's a temporary thing, but spontaneous ovarian hyperstimulation syndrome and precocious puberty are both in that category. So I do have one question. Which of the following reproductive disorders gives the greatest chance of having a DNA diagnosis? A, hypogonadotropic hypogonadism, B, polycystic ovary syndrome, C, Mayer-Rokitansky-Kuster-Hauser syndrome, D, endometriosis, E, hypergonadotropic hypogonadism. So this is something to think about and we'll answer this at the end of the talk. So I'm going to briefly review the hypothalamic pituitary gonadal axis. It's probably familiar to most people here. In the hypothalamus, you have pulsatile GnRH is secreted, which stimulates the pituitary to secrete FSH and LH, and then they stimulate the gonads to secrete sex steroids and gametes. There's also negative feedback from estrogen, in this case, estrogen and progesterone, at both the hypothalamus and pituitary. And importantly, GnRH neurons, as was discussed by Dr. Krauss, they originate outside the brain. And as shown in the mouse here on the left, in the yellow, is the olfactory placoid region where the olfactory nerve migrates across the cribriform plate and goes to the hypothalamus. And as shown up here in a little more detail, the olfactory epithelium, you can see the olfactory nerves going across the cribriform plate. The little circles are GnRH neurons, and they travel along with olfactory nerves to get to the hypothalamus, thereby explaining what can happen if you disrupt both of those disorders. You get disorders of smell and reproduction. All right, when someone is hypogonadal, we don't know where the etiology is. It could be in the hypothalamus, it could be the pituitary, or in the gonad. So you have to assess this. And so what happens when sex steroids are low, the negative feedback to the brain is abolished. And so the way we tell the difference if it's hypothalamic pituitary versus gonadal is we draw FSH and LH levels. And this really should be done in men and women. And this way you can categorize patients into the hypothalamic pituitary, which you got to consider an MRI to rule out a tumor, and if it's gonadal, if gonadotropins are high. So what I'm going to do is start with hypogonadotropic hypogonadism, and it's impossible to discuss every gene. I'm going to give the first Calman syndrome gene, the first normosmic gene, and then several highlights, and then concepts. And then the same will be true with hypergonadotropic hypogonadism. So in hypogonadotropic hypogonadism, here's the reproductive axis shown again. You have sex steroids are low. There's no negative feedback to the brain. And so when we draw FSH and LH, they're low, or they're inappropriately normal, which is not normal in the hypogonadal state. So this indicates a hypothalamic or a pituitary defect. So there are various prevalences in the literature. I'm not sure which is the actual one. It's interesting. It's always said it's much more common in males, yet almost all the genes are autosomal. So I'm not exactly sure how to reconcile that. But in general, it's defined as irreversible delayed puberty. That's classically. There are exceptions to that. In females, usually by age 17, who have hypoestrogenic amenorrhea, and males age 18 with low testosterone. Gonadotropins are low or inappropriately normal. There's no pituitary tumor by MRI, and other pituitary function is normal. Calman syndrome then can, couples anosmia with hypogonadotropic hypogonadism. And what's become known over the last 25 years is there are a number of other associated anomalies which make counseling incredibly important because some of them are fairly major. They're midline facial defects, a variety of neurologic defects, deafness, renal agenesis, cardiac anomalies. So the first gene identified was the Calman syndrome gene, or CAL1, encoding anosmin-1. And you would expect that this gene would encode a protein that affects migration, and mutations should result in both. And that's actually what happened. Several groups described this, and this was the first gene identified and is X-linked recessive affecting males. And virtually all males affected have anosmia, hyposmia. This is a good example where the understanding the expression patterns correlates very nicely with the clinical findings. So you can see the areas here in the olfactory bulb that correlates with anosmia, some of the neurologic abnormalities in the cerebellum and spinal cord. It's also expressed in the meso and metanephros, which goes with renal agenesis, and in the facial mesenchyme, causing midline facial defects. And these have been known for quite a while. The first autosomal gene to cause hypogonadotropic hypogonadism was the Gnrh receptor. And our group and a French group, about the same time, our paper was in January of '98, theirs was in November of '97. They studied a family with partial IHH, and they found mutations in the Gnrh receptor. We screened both normosmic and anosmic. Nature Genetics made us get rid of the anosmics because they said they were all due to CAL1 mutations. But we found a mutation in one family, and one of the patients had been given Gnrh and was markedly resistant. And this is the family that I took care of when I was a fellow, and you can see that the three females were all fairly old when they were diagnosed, and none of them had any breast development. So age 30, 21, and 17, no breasts. Their gonadotropins were low or normal, and the only male affected had a very low testosterone. In this assay, less 300 nanograms per DL was considered low range and normal at this time. He would not permit a testis exam, but he had never had a voice change. So these patients all had compound heterozygous variants in the Gnrh receptor, which in collaboration with Jimmy Neal showed impaired signal transduction. And this shows the G protein coupled receptor, the Gnrh receptor, with 7 transmembrane domains, an extracellular domain, and then intracytoplasmic and extracellular loops. And interestingly, there's very small or absent carboxy terminus. But both of these variants affected signaling in vitro. And if you look at this, this was the first gene identified in normosmic IHH. It was autosomal recessive. And the mutations they can cause mild to severe phenotypes. Usually there are no other anomalies with Gnrh receptor mutations, and many of these are compound heterozygous missense mutations that affect ligand binding and/or signaling. And they were relatively frequent in the normosmic population of 4 to 5%. Now we know there are a large number of genes, probably 34, 35 or more, and this is some of them. I've overlaid it in the hypothalamic pituitary gonadal axis, and as you would expect, most of these affect hypothalamic function. However, NR0B1, which encodes DAX1, has effects at both the hypothalamus and pituitary, and then there are at least 6 genes, more than this, that cause combined pituitary hormone deficiency, and these are largely homeobox genes, so we have to keep those in mind as well. So about 40% of the cases now, the molecular genetics is identified. Digenic has been suggested anywhere from 2 to 20%, and that makes counseling challenging, although proving digenic disease is very difficult, and it's very difficult to get funding to prove it too. So if you look at there's some generalizations we can make regarding some of these genes. COL1 and FGF8 bind the FGFR1 receptor, and then there are other genes also, the ligand and receptor. These happen to be involved in GnRH neuron migration. Then you have the GnRH system, leptin and its receptor, kisspeptin, neurokinin B. These seem to affect mostly GnRH secretion. And interestingly, TAC3 encodes neurokinin B, and kisspeptin is also included in these so-called candy neurons that have kisspeptin, neurokinin B, and dynorphin, and they regulate GnRH neurons. So the mutation phenotype can be very different. So what I've told you so far, with COL1, they tend to have Calman syndrome. Normosmic IHH, some examples are shown here that we just discussed, but there are some where you can have mutations in the gene and they can cause either Calman syndrome or normosmic hypogonadotrophic hypogonadism. So they must affect GnRH neuron migration and/or regulation. Some of the associated anomalies I'll show here. Calman syndrome we already discussed. With FGFR1, you can have cleft lip and palate dental agenesis, synkinesia. In leptin and leptin receptor, hyperphagia and morbid obesity. NR0B1 is DAX1, that's congenital adrenal hypoplasia. CHD7, it's CHARGE syndrome, and we recently, with collaboration from Sue Kim in London, found WDR11 is probably a ciliopathy. CHARGE syndrome is well known to this audience with all the abnormalities listed here: coloboma of the eye, heart defects, posterior nasopharyngeal abnormalities, retardation in growth and development, and genital anomalies, and ear abnormalities. We had hypothesized and showed that IHH Calman syndrome is a mild allelic variant of CHARGE syndrome, and we found about 6% of our patients with normosmic hypogonadotrophic hypogonadism and Calman syndrome without full CHARGE features had CHD7 mutations. Very interestingly, different effects different phenotypes can result depending on the type of mutation. As it's well known, FGFR1 mutations, if they're activating, can cause craniosynostosis with a variety of different disorders, and inactivating mutations cause Calman syndrome. And as we've heard at this meeting, now there's several genes where you can actually maybe have the phenotype of both with a mutation. Other associated anomalies that have been found: SOX10 is very high prevalence of deafness. There are at least 3 genes that cause ataxia, and the Gordon-Holmes syndrome is shown here. In particular, neurokinin B and its receptor, these are the patients that, if you have mutations in these genes, tend to be the reversible hypogonadotrophic hypogonadism. So those that are treated with either gonadotropins or testosterone, you stop the therapy and their gonadal function resumes. SOX2 can cause combined pituitary hormone deficiency, eye abnormalities here, hearing abnormalities, and septo-optic dysplasia. LHX4 causes combined pituitary hormone deficiency, isolated growth hormone deficiency, and then smaller ectopic pituitary glands. So looking at a lot of these genes, you can see there are several at least that are X-linked recessive. Many I've categorized here as biallelic or autosomal recessive, or heterozygous or autosomal dominant, because they're not large family studies. We're presuming these are autosomal dominant or autosomal recessive, but it's still not clear. And the digenic issue is also a problem. If you look at what are the common ones, You can see in Calman syndrome, 3 genes are relatively common, and in normosmic hypogonadotropic hypogonadism, 4 are. And in general, FGFR1 and CHD7 tend to be some of the more common causes of Calman syndrome. But there are at least 34 genes known right now that cause Calman syndrome. Now, the digenic issue is perplexing. When we published this paper in 2011, the ones in blue are the cases we published, and then we looked at all the ones in the literature up to that point. And in parentheses, the number of alleles affected are shown. And the thing that's interesting is 1 gene is sufficient to cause it in many of these cases. In fact, 15 of 18. Look at example particular is number 8. Heterozygous FGFR1 has clearly been shown to cause it, as well as biallelic Gnrh receptor mutations. So it's not clear yet how this will all play out, but digenic diseases in general require mouse, human, and cell studies, but they're very difficult to convince study sections to give you money for it. For pituitary deficiency, I mentioned there are at least 6 genes here shown that cause combined pituitary hormone deficiency. And the reason I put this in is to remind us when we see these patients, you do have to make sure they don't have pan-hypopituitarism. Most patients we see with presumed Calman syndrome or hypogonadotropic hypogonadism have hypothalamic, but hypopituitarism can be a major medical issue for the patient. I'm going to mention one interesting rare disorder is isolated FSH deficiency. And one thing you need to know regarding this phenotype first is that LH and FSH are dimers. They have a common alpha subunit, but the beta subunit specifies their function. So in these patients have low levels of sex steroids, so you have negative feedback and is blocked for FSH, but LH is quite high, and FSH does not rise because there's mutations in the FSH beta gene. And there are not a huge number of these patients. Several groups have described these in the past. It's autosomal recessive, and these are the females, at least 6 of them all had primary amenorrhea, so they never had a period. A lot of them didn't have any breast development, or they had partial. And interestingly, the 3 males were all azoospermic, and one of them had delayed puberty. But all of the FSH beta mutations we studied with collaborators at Mass General had decreased immuno and bioactive FSH. And what's very interesting is we studied this patient and drew her LH levels in the Clinical Research Center when I was at the University of Chicago, and her pattern of LH secretion resembles a PCOS patient. In polycystic ovary syndrome, they have very high LH, they're hyperandrogenic, and interestingly, this patient had no hirsutism and her testosterone was low, indicating that FSH is also important in androgens. I'm going to turn to hypergonadotropic hypogonadism. And in this case, here's the reproductive axis again. So you have low levels of estrogen, negative feedback is abolished. So now you draw FSH and LH and they're elevated. So this means that there's a gonadal defect. So one we always have to remember is Turner syndrome, and it's more common in primary amenorrhea, but still in patients who have secondary amenorrhea, you have to keep it in mind. But 25% of cases of primary amenorrhea and a little under 1% of secondary amenorrhea can have Turner mosaicism or pure. But as well known to this audience, short stature is the most constant feature. And it's often reported that patients have puberty. 10% of 45X and 50% of mosaics have puberty, but usually the menses are very short-lived and there's major ascertainment biases in many of the publications. But we must exclude a 46XY cell line in these patients because of dysgenetic gonads, and then all of the other associated anomalies well known to this audience, so cardiac anomalies, renal anomalies. And as a reproductive endocrinologist, we're very hesitant and try to dissuade people from doing donor egg IVF because of the potential risk of an aortic rupture.
If you look at the genes for 46XX, hypergonadotrophic hypogonadism, the two of the prominent ones, FMR1, which we'll talk about, and FSH receptor was one of the earliest ones identified. But there's a variety of different mutations that are known, and now about 20 to 25% of cases of women who present with primary amenorrhea who have elevated gonadotropins will have a mutation in one of these genes. So if you look at the phenotype, it can be isolated. Premature ovarian insufficiency is the newer term instead of ovarian failure. And there's some genes listed here, BMP15, FSH receptor, FIGLA, and NOBOX. And then there are a number with other anomalies, and then there's some uncertain ones. And if you look at the ones with anomalies, Fragile X syndrome is well known to this audience. As you know, some of the females can have tremor and ataxia. With 17-hydroxylase deficiency, or CYP17A1, you can have adrenal insufficiency. With aromatase insufficiency, or CYP19A1, they can have sexual ambiguity and ovarian cysts. Mutations in SF1, the gene NR5A1, which encodes SF1, regulates steroidogenesis in the adrenal and HPG axis. And then there are several genes that cause leukodystrophy as well. GALT gene is known to cause galactosemia. Autoimmune polyglandular syndrome type 1. And one to me that's incredibly interesting is FOXL2, which mutations cause blepharophimosis, ptosis epicanthus syndrome is shown here. Very interesting that eyelid abnormalities and ovarian insufficiency would go together, and a pretty uncommon situation, but one thing that's very important is this FOXL2 is now known to be a key determinant, ovarian determining gene. So from these obscure, rare phenotypes, you can find major contributions to normal physiology. And then more recently, Alex Rakovic's group has found these genes MCM8 and MCM9 by whole exome sequencing. These are autosomal recessive disorders, short stature, and chromosomal instability. And in fact, he has recently published this list. As you can see, these are all autosomal recessive. They're all pretty uncommon. They're about 1% prevalence. But many of these affect cell division or apoptosis, meiotic recombination. So there's a theme in male and female infertility of having some of these processes affected. So if you look at the inheritance of these, by far autosomal recessive are the most common, but there are some X-linked dominant, FMR1 and BMP15, and then there's some that are uncertain as well. Fragile X syndrome I'll just briefly review because of its importance. It's X-linked dominant disorder with incomplete penetrance, and the male phenotype is well known of intellectual disability and some other abnormalities. And due to expansion of a triplet CGG repeat in the 5' untranslated region of FMR1. And so it's the pre-mutation carriers that are at risk for having this ovarian insufficiency. The full mutation over 230 repeats is a null mutation with reduced protein. The FMR1 promoter is methylated and inactivated, but that's not associated with premature ovarian insufficiency, whereas the pre-mutation is. And older males and some females with pre-mutation get the tremor ataxia, but this is an RNA overexpression mechanism, not protein reduction. And it's been quite a while ago that this has been nicely defined in large Fragile X families, showing about a 16% prevalence of premature ovarian insufficiency in Fragile X carriers, pre-mutation carriers. You can see none of the full mutation patients had it. If you look now at women with premature ovarian insufficiency, if it's sporadic, there's about a 3 to 4% chance that the woman will carry a pre-mutation. If she has a sister or another relative, it goes up to 12 to 15%. We had an interesting patient a few years ago who presented at age 15 to the pediatrician with lower abdominal pain. Her ultrasound showed large cystic ovaries, and she was referred to a reproductive endocrinologist and pediatric adolescent person. He noticed absent breast development and ordered some laboratory studies, and this is her exam. She was thin, she had no breast development. This was at age 18, no abdominal mass, and had mild acne. Her initial lab showed an estradiol of 3,500 picograms per ml, which pre-ovulatory, if you have one follicle, it's 200 to 300. So this is like an IVF hyperstimulation patient. Her gonadotropins were mildly elevated, and she had complete estrogen insensitivity. So we sequenced the ESR1 gene, which encodes estrogen receptor alpha, but her growth, her linear growth was normal. Her weight is on the low side, as you can see at the bottom left. But what's really interesting is the steroid-dependent growth spurt was absent in this patient. And so we sequenced the gene. She had a homozygous deleterious missense mutation that was completely conserved in every species we looked at. It impaired signaling 240-fold. Unfortunately, she was adopted. So we did an array, and the array showed an 11% region of homozygosity. So her parents were second-degree relatives and likely heterozygotes. So this was the first female who was reported with estrogen resistance. It had been almost 20 years previously that a male was described, and it was thought that they would be lethal in females. So there, subsequently, another paper came a few years ago with 3 SIBs, 2 females and a male. They had similar stories of high estradiol. The male had cryptorchidism and a low testosterone, and his estradiol was elevated too, and had a missense variant in the ligand binding domain. These were homozygous also, and they impaired signaling 65-fold. So how common are these ESR1 variants? As we know, androgen receptors, there are hundreds of mutations. The estrogen receptor shouldn't be privileged. So we're looking at this right now. I'm going to turn my attention to the eugonadal category. I have spontaneous ovarian hyperstimulation and precocious puberty asterisks here as hypergonadal, as a kind of a temporary state, but I'm going to go through these. So ovarian hyperstimulation is usually induced by us. These are women having infertility treatment with exogenous gonadotropins, and normally you should have one follicle. This is showing 2 ovaries, markedly enlarged ovaries that can cause pelvic pain. You can have third spacing fluid, and it can be life-threatening. However, it can happen spontaneously without any treatment. And what's been found is we know that inactivating mutations in the FSH receptor, as I told you, cause hypergonadotropic hypogonadism, but activating mutations cause this entity of spontaneous ovarian hyperstimulation. And there have been at least 5 females described with this. 2 of them are familial. They're heterozygous mutations. 3 of the 5 FSH receptor variants were constitutively active. And the reason this only manifests in pregnancy is because HCG goes up. So these patients, so HCG or TSH can stimulate this receptor when it has mutations. And here's just one of the families showing here. The numbers refer to the number of pregnancies that were affected. So the proband here had 5 pregnancies, and 4 of the 5 were affected. She had ascites and ovarian masses and had a therapeutic abortion at 8 weeks. Then she had a spontaneous abortion with no ovarian hyperstimulation. Then she had a couple more pregnancies, and she went to term, but had ovarian hyperstimulation the entire pregnancy. And interestingly, all 3, and her and her 2 sisters both had heterozygous FSH receptor mutations. So it may not affect every pregnancy, but in general, it's an impressive entity. I'm going to mention precocious puberty briefly. Precocious puberty in girls is usually puberty before age 8, and boys it's under age 9. And it can be central, where you activate the HPG axis. Or it can be peripheral, having a tumor, for example, of the adrenal or ovary. So Ursula Kaiser's group at Brigham did whole exome sequencing in 15 families, and they found 4 heterozygous variants in this McCoran ring finger protein 3, or RN3 that were paternally inherited. And interestingly, this is in the Prader-Willi critical region on 15q. And she found that 4 out of 15, or 27% in this study, had mutations in this gene, and it's autosomal dominant. And it's now thought to be about 40% of kids with central precocious puberty, so that's fairly common. I'm going to go through a couple rare causes of eugonadal infertility. This is just showing a normal follicle. You can see the egg in the middle, the oocyte, the zona pellucida is the covering around the egg, and then the granulosa cells and then the thecal cells both nourish and communicate with the egg, and especially the granulosa cells that surround the egg, the cumulus. So the zona protects the egg and facilitates crosstalk between the cumulus cells and the egg so the egg can mature. There are 4 different ZP proteins, ZP1, 2, 3, and 4, and recently in a consanguineous Han Chinese family, there was some infertility, and very interestingly, these patients had IVF and had no zona pellucida. They didn't get pregnant either. So they sequenced ZP1, 2, 3, and 4, and they found that all of these patients that were studied were homozygous for a ZP1 frame shift variant. And this variant affected function. I'll show you here. So here's a normal oocyte, and you can see the egg right here, and the zona pellucida right surrounding it. And then if you look at the interaction, this is the way the different ZP proteins aggregate to cause the normal zona. But in the patient with the mutation, there's no zona pellucida around the egg, and when they looked at the function of the protein, it just aggregated in the cell and didn't make it out. So they clearly affected function. Interestingly, ZP3, there's also been mutation in that gene as well, and it causes what we call empty follicle syndrome. You do IVF, you go in to get eggs, there are no eggs. And they did it by genome-wide linkage and whole exome sequencing. They found heterozygous paternally transmitted ZP3 missense variants in a couple different families. It interfered with interaction of ZP2 and ZP3, so interfering with the crosstalk of the egg and the cumulus. And interestingly, they studied it in another cohort, and it's roughly about 10% prevalence in these patients. There have also been some other mutations affecting oocyte maturation and zygote cleavage as well, and these genes shown here, PAT-L2, TLE6, and PAD16. Mullerian aplasia is a perplexing disorder, congenital absence of the uterus and vagina, or Mayer-Rokitansky-Kuster-Hauser syndrome, which the patients prefer to be called. It's the most severe Mullerian anomaly, affects 1 in 4 to 5,000 women, constitutes 10% of the cases of primary amenorrhea, and has a variety of other anomalies associated, renal, skeletal, cardiac, and deafness. If you're looking carefully at the literature, there's pretty good evidence for 2 genes, Wnt4. There's several mutations, and some of these patients have hyperandrogenism as well. And HNF1B, there's an intragenic deletion and maturity onset diabetes of the young with renal cysts. We've sequenced more than 100 people, both of these genes, and found nothing. So I think they're fairly rare. There have been a large number of CNVs reported and other genes associated, but they're not clearly causative at this time. Now I'll finish in the final few minutes with a couple more eugonadal common disorders, and association studies have been used to study these. So these are things like PCOS and endometriosis, and I think this audience knows this very well, but I mean, an association study is a case control study comparing the frequency of a polymorphism in the study group versus the control. And they can be any kind of SNPs or CNVs or repeats. You perform a test of significance. It's actually better if you have some family members as internal controls. Now they're usually done as genome-wide association studies. And you need usually a primary study and then a replication study to reduce the noise. Remember that polymorphism, reasons for association, I mean, polymorphisms can cause the disorder. That's incredibly uncommon. but they can also, they don't cause the disease, but they can be in linkage disequilibrium with the disease-producing gene. Or sometimes they're more frequent in controls because of artifacts of population admixture and by chance alone. So association studies are not the same as causative studies, although they're incredibly important for dissecting the biological basis. So I'm going to talk briefly about three entities: endometriosis, which is endometrial glands and stroma outside the uterus. You can see the glands and the stroma here. This is in an area of the peritoneum. And these patients present clinically with pain and/or infertility, and they're diagnosed by laparoscopy. Endometriosis can have a variety of appearances. Some of them are shown here. They can be blebs or powder burned or cause scarring or purple areas. And, or they can be even more severe, where they encase the whole ovary, as an ovarian endometrioma here, or cause adhesions in a tubo-ovarian complex, as shown here. At this time, there have been association studies, and there are some loci associated and some that have been replicated. But at this time, there's no causative gene that we can use in clinical genetic testing for our patients with endometriosis. Polycystic ovary syndrome is the most common cause of anovulation, and it leads to infertility. The Rotterdam criteria are usually used for diagnosis. You have to have 2 of 3, either hyperandrogenism, hirsutism, anovulation, or polycystic appearing ovaries, and a polycystic appearing ovary is shown over on the side. Many small follicles, because they don't ovulate often, and you must exclude other causes. We worry about endometrial cancer and the risk of diabetes in these patients. There have been a huge number of association studies, and the more recent ones have suggested DENN-D1A, FSH receptor, LH receptor, and some other genes. And then there are several loci that have been confirmed with replication as well. But at this time, there's not a causative gene that we can use for clinical testing. I still suspect at least a small percent of these patients will have Mendelian causes, just like in hypertension. Fibroids is the last one I'll mention. These are smooth muscle tumors that arise from myometrial cells. They cause bleeding, pain, and infertility. It's the most common reason for a hysterectomy. And you can see the locations can vary inside the cavity, within the muscle. They can be pedunculated or outside. And you can see a very large uterus here with multiple fibroids. And here's a pathologic specimen. So there are a number of associated loci. But what's interesting is actually the somatic mutations in tumors. About 75% of people and 75% of tumors have somatic MED12 variants. And it's interesting because in the germline, MED12 mutations cause intellectual disability in males. So that's kind of an interesting, but there's no causative gene to now test women for fibroids either. So in summary, the genetic etiology of hypogonadism, the more severe phenotype, is much better known than the eugonadal. And about 40% of germline mutations, digenic 2 to 10%. Got to rule out pituitary insufficiency and make sure are there associated anomalies and counsel patients with recurrence risk. For hypergonadotropic hypogonadism, we still have to rule out a 45X cell line and a 46XY cell line. And then if they're 46XX, fragile X is the most important thing to consider. For these other disorders, ovarian hyperstimulation, familial central precocious puberty, and the miscellaneous rare eugonadal disorders, it's worth sequencing the gene in those patients. For Mullerian Aplasia, there are only a couple genes that make up a couple percent. And for endometriosis, Pcos, and fibroids, there's really not good evidence for germline mutations for causation. So what are we supposed to do? I think in hypogonadotropic hypogonadism, you could base it on the phenotype. I mean, if they have deafness, going for SOX10 is very reasonable. I showed you the prevalence of some of the more common ones. Testing could be based upon anomalies, but also targeted next-generation sequencing is probably the most beneficial. But you do have to be careful with the digenic issue. For hypergonadotropic hypogonadism, after excluding a 45X or 46XY cell line,
Fragile X testing is indicated, and then you could consider targeted NGS for the other variants. And as I mentioned, these others were the ovarian hyperstimulation, precocious puberty, empty follicle syndrome, and these other ones. You can sequence the known gene here. However, for Mullerian aplasia, endometriosis, PCOS, and fibroids, testing is not terribly practical at this time. So my question is, which of the following reproductive disorders gives the greatest chance for DNA diagnosis? So what do people think? So it's A. So I think I'll stop there. Thank you for your attention.